The overall goal of qPCRTag analysis is to improve the throughput of the Synthetic Yeast Genome Project PCRTag Genotyping Assay by implementing a real-time PCR-based detection system and miniaturized reactions. This is accomplished by first using a bulk liquid dispenser to distribute qPCR master mix into each well of a 1536 multi-well plate. Next, a nanoscale acoustic droplet ejection robot transfers template DNA and primers into the wells of the multi-well plate. Then, the plate is sealed, and the reactions are run through a qPCR thermocycler. Finally, the real-time PCR data is analyzed to determine the presence or absence of synthetic or wild-type PCR tags at each locus. Ultimately, the qPCRTag workflow improves throughput.
The main advantage of this technique over the current PCRTag assay is that it allows us to circumvent the use of gel electrophoresis and manual annotation of those gels.
We first had the idea for this method when we realized how time-consuming and labor-intensive PCR tagging on longer chromosomes was going to be.
After culturing and harvesting approximately 2 million yeast cells, add 200 microliters of yeast lysis buffer and pipette to resuspend the pellet. Add 300 microliters of acid-washed glass beads to approximately 1 millimeter below the level of the cell slurry. Then, in a fume hood,